As we mentioned in the introduction, my lab works primarily on the human microbiome and other microbial communities, some for methods development, some for analyses. And the human microbiome in particular, as many of you are familiar with, has been of interest for the past several years now for, I would say, two main reasons. One is primarily technological. As we'll see over the next few slides, high-throughput sequencing in particular has given us a new tool that allows us to see the human microbiome and other microbial communities in much greater detail and much cheaper and more conveniently than we could with previous tools. But second, I know there are a lot of people here who think about genomics or who think about transcriptional regulation. One of the biological models that I like to think about for the human microbiome is as a really remarkable supplementary set of differentiated cells that we carry around with us. So you have trillions of microbial cells hosted in your gut, elsewhere on the body. They carry up to around 1 million genes per individual. So you can think of this as a supplemental genetic repertoire that is more extensive in some ways and certainly much more plastic than the human genome. These bugs are able to turn over and modify their genomes very quickly in interesting ways. And from a regulatory standpoint, which operates on a very interesting time scale as far as regulation. If you think about your microbes as regulating their activity by growing, by doubling, or by dying off, that gives you a time scale of regulation that operates on, say, hours to days, which is relatively unique relative to human molecular biology. We can make transcriptional changes very quickly, genetic changes very slowly, maybe epigenetics somewhere in the middle, but our microbial communities give our larger biological system a really neat type of regulation to play with. So to talk about how we can now see these microbial communities a little bit, unsurprisingly, like many, many other things in biology right now, cheap, convenient, high-throughput sequencing has been the technology that's unlocked our ability to study the human microbiome and other microbial communities. And to give a quick overview of how sequencing can be a tool to study communities, everything starts with a culture-independent microbial sample. So this can be going back about 10 or 20 years when a lot of this work started, often environmental, soil or water, where you scoop up a sample and check out which microbes are in it. Or for the human microbiome, maybe some less pleasant sample types, but you're starting with a swab or a collection of microbes that you want to crack open and extract the nucleotides from. Typically, this is still DNA specifically. Increasingly, there's work in microbial community transcriptomics, in which case you'll extract RNA instead. And there are two primary paths by which sequencing is currently used for these types of nucleotides. Either amplicon sequencing, in which a single gene is targeted for amplification out of the whole mixed bag of microbial DNA. Very commonly, this is the 16S RNA subunit gene, so you'll see this referred to as 16S sequencing. Increasingly for fungal studies, you'll see other amplicons used, either the 18S ribosomal subunit or internal transcribed spacers, or ITS amplicons. In any of these cases, what you're doing in amplicon sequencing is picking your one target gene, amplifying out just the variants in that gene across different members of your population, sequencing just those sequences, and classifying each sequence variant to its microbial source. So this is a way of performing taxonomic profiling. You treat each sequence variant as a name tag. You can count up, I see 5 sequences that came from the first microbe, 10 sequences that came from the second microbe. And what this provides is data that looks something like this, where for each microbial community sample, you have a list of which microbes were present in the community, how many sequences you classified to their particular name tag, and a set of counts or relative abundances that tell you which microbes were there. So again, you'll see this referred to as 16S or amplicon, more generally, sequencing. This is still the cheapest and easiest way to get information on a microbial population census, to tell you which bugs are present. Although the difference in price between this very targeted sequencing and more general metagenomic or metatranscriptomic sequencing has been and continues to come down. So when you take the extracted DNA or RNA and sequence the whole shebang, this is again referred to for DNA as metagenomics or RNA as metatranscriptomics, and it generates exactly what you'd expect for a single genome sequencing, big pile of short or as we just heard, increasingly longer reads depending on your technology. This is great in that it can provide you a much more complete picture of a microbial community. You get not just single genes that serve as name tags, but a little bit of everything. And it's good in some ways and bad in others in that it's also much more complex data. So this can tell you with the right kind of computational tools, not just about the relative abundances or the taxonomic or profile or census of which microbes are there, but what their gene content looks like, what their genome structure looks like, what their genetic variants, if you have enough sequencing data, look like, or some of the areas that we're interested in, what their metabolic or functional profiles look like, what are the functional capacities of the combination of microbes in community. So both of these paths through high-throughput sequencing to study microbial communities show up increasingly on the right-hand side. There's a tremendous amount of data out there now for the human microbiome and other communities on the left-hand side for amplicon sequencing. A lot of what I'll talk about today is recent analyses that we've done using shotgun metagenomics or metatranscriptomics and some of the methods development that have made interpreting that picture a little more possible. So the three main types of analyses that get carried out from shotgun metagenomics and metatranscriptomics in particular, I would classify in three areas. One, again, taxonomic profiling. If you have shotgun data, you often still want to know which microbes are present. And there are some more specific or a little more nuanced ways in which you can do this rather than just taking single sequence amplicons as you might for 16S sequencing and classifying each one to a microbe. And I'll talk a little bit about methods for this today. Second, you might be interested in functional profiling. What are the genes or pathways or metabolic capacities that are present in a community rather than what are the bugs that are present there? And the one picture overview that you can think about this as classifying each read of interest into a gene or into a pathway, counting up which genes or pathways you see instead, and treating those as counts or relative abundances in your samples. And I'll talk a little bit about methods and results for this as well. The third major path through here is assembly, and fortunately we just heard a lot about that because we don't do a lot of work here. It's incredibly challenging to do metagenome assembly. Take everything that you just heard for single genomes and multiply that by 100 or 1,000. As Mike said towards the end of the talk, this is really a big next step as far as understanding microbial populations. So what I'll talk about is mainly reference-based assembly-free methods for taxonomic or functional profiling in shotgun metagenomes, in shotgun metatranscriptomes, and some applications of those to human health. So in order to do these types of analyses in an assembly-free manner, we work a lot with the huge database of reference information that's out there now for bacteria, for archaea, and for other microbes. And a lot of this work goes back to a former postdoc in the lab, Nicola Sagata, who now is his own lab at the University of Trento, who had the intuition that we can really use all of the microbial isolate genomes that are being deposited, also with incredibly accelerating frequency, as a reference, as a source of information about microbial populations in the wild. So this slide is one of those where no matter how often I update it, it's always out of date, it's always wrong. These numbers are reasonable approximations at this point, but there are somewhere around 20,000 bacterial-specific reference genomes at the NCBI right now. And these range, as we just heard, anywhere from beautiful, high-quality finished genomes to collections of contigs, all of which are useful as reference information for interpreting microbial populations. These are in addition to a few thousand viral genomes, which are also have their own challenges in assembly and interpretation, and a few hundred each of archaeal and eukaryotic microbial genomes. Oh, this is the increase up to the last time I updated the numbers in the number of annually deposited microbial genomes. It's up to the point where we're exceeding about 1,000 per month right now. So I don't have the graph going out that far, but there are huge numbers of microbial genomes being deposited. So there are projects that will sequence, say, 1,000 different streptococci in one big go and deposit those genomes. So that's why the numbers are always a little wrong. There's a reasonable amount of phylogenetic diversity in the current reference set. There are great projects like GIBA that are working to expand that. But as of a few months ago, this went out to just over 4,000 species and about 50 phyla. Unfortunately, the phylum number has stayed relatively constant, but we're continuing to expand our species level coverage and genera and even up to the class level or so very, very quickly. For the purposes of functional interpretation of microbial communities, one of the things that I get excited about are not so much the genomes themselves, but the genes that have been annotated within them. So as we'll see in a couple of examples today, there are about 55 million unique gene families among these deposited genomes that can be used to interpret the functional content or the functional potential of microbial communities. And we can take these genes and catalog them and organize them. And this is what Nicola has done in a database called Chacoflan. That's a catalog of orthologous genes that can then be used for interpretation of microbial communities. I'll talk a little bit today about how the unique gene sequences among microbes in this catalog can be used to tease them apart and identify who's there for taxonomic profiling in communities. I'll talk a little bit about how to use the functional annotations on these genes for metabolic profiling of communities. And I won't have time today, but Nicola's also done some work on using the most conserved, the least unique sequences to build higher quality microbial phylogenies as well. So to start with taxonomic profiling, that first task that we wanted to be able to do with shotgun metagenomic data, to identify who's there in a community. A system that Nicola put together initially a few years ago called Metaflan uses the concept of unique marker sequences to identify which bugs are in a community. And a unique marker sequence is a special type of core gene where a core gene is conserved within some level of sequencing and assembly and annotation error throughout an entire clade of interest. So a species-level core gene would appear in all or almost all of the isolate genomes from that species, and possibly elsewhere as well. A unique marker sequence is a core gene that only, again, within some degree of allowable error, that only appears within that particular clade. So if you see, for example, a unique marker sequence from a particular microbial species in a community, you have very strong evidence that that read that maps to one of those unique sequences came from that microbe, and you can count them up for taxonomic profiling. So,
This might be very ugly, or you're not going to talk about it, but how do you decide that two different sequences are actually different genes?
as far as the
Variance do you need? So how much sequence divergence do you need between two different genes, or just two genes that are mutated from a gene that is mutated?
So the question was how much of a homology do we require to say that two sequences are the same gene? Is that a fair paraphrasing? We typically use two cutoffs, one that's the specific threshold for species-level gene families at 97% identity, or 97% full-length identity. And then to build a larger protein-level catalog that we use for other purposes, we use an 80% nucleotide-level homology cutoff. Yeah, because we can go back afterwards. I won't go into the details of building the catalog today, but it's a hierarchical process. So it's very easy to start at the bottom, be very stringent, then back off to do, say, a family-level 80% nucleotide homology clustering, then back off again and do, say, an 80% amino acid level homology clustering if we want to make a more general catalog out of the base. So the short answer is 97%. Other questions at this point?
Do those apply to bacterial genes as well? Those cutoffs?
Those are for, the question is whether the cutoffs apply to bacterial genes,
Sorry,
viral genes. We build our viral markers a little bit differently, and I'll mention that in just a second here. So the pipeline that Metaflan, which is our metagenome taxonomic profiler, uses to take advantage of the Chacoflan gene family catalog is to grab out the unique marker genes for each possible clade of interest. And this might be a species or a genus or a class. Pre-build that reduced targeted catalog in which we know with high confidence the taxonomic origin of each sequence, and given a new metagenome, map short reads just to that unique marker database so that every hit is informative about the taxon of origin. And we can then count hits and build a taxonomic relative abundance profile just like you might get from classification of amplicon sequences. We've just recently finished up Metaflan 2, which is an update of the original Metaflan system. We target about 200 marker sequences per species when possible. This is in contrast to the single marker sequence that you get from amplicon sequencing. So we get a great boost both to taxonomic specificity. It's very easy to tell species apart using these repeated measures, and to detection sensitivity and specificity. We have false positive and false negative rates around 1 in a million at this point. So given enough sequencing depth, and this is for about 2 gigabases of sequencing. So given enough sequencing depth, you can get very sensitive and specific taxonomic profiling from shotgun sequencing to parallel what you might get from amplicon sequencing. The major updates in MetaFlan 2 are to include, rather than just bacteria and archaeal markers, viral and eukaryotic marker sequences as well. Eukaryotic markers are derived in about the same way, and to now answer the earlier question about viral markers, because there is both sort of ambiguity in the reference databases for viral sequences and a lot of honest-to-goodness biological sequence sharing between, especially, for example, phage and bacteria, what Nicola does to build viral markers is to use entire viral genomes and require, for example, hits to unique regions of the genome or to essentially breakpoint regions of the genome. We can perform strain-level profiling in addition to species-level profiling of microbial communities, which I'll show in a couple of slides, using combinations of markers rather than marker averages. And much like isoform disambiguation in RNA-seq, we can use quasi-markers or slightly ambiguous markers for which we might not have a unique mapping, but which through expectation maximization we can tease apart what the more likely taxon of origin was later on in the processing pipeline. And putting this together, what it ends up looking like for an example of a MetaFlan 2 derived taxonomic profile from a series in each column of metagenomes from a set of skin samples, combining the Human Microbiome Project with a cohort that Nicola is working with locally in Italy, is for each row, we get a readout of the species-level relative abundance, both of some of the major skin bacteria. So if you look at the 3 brightest lines here, we see Propiacnes, Staph epidermis, and Staphylococcus, a different one, Caprolcapitis at the top here. So 3 of the major bacterial skin colonists. But if you squint real hard and you recognize some of the microbiology, Malassezia is the dominant skin fungus in healthy populations, so we're able to pick out eukaryotic microbes. And scattered throughout here are a small number of viral detections, typically phage, that do at the population level correlate with their known hosts. So for example, you can see in these 2 propiphage examples at the bottom, they correlate to a high degree with propy abundance, and they actually tend to exclude each other. So you have one propiphage but not the other in this particular example. But we can keep zooming in with MetaFlan 2 and provide strain-level tracking as well. And this is done through something that will be important for the next example of microbial community profiling from the lab. To a first approximation, we all have a different strain of everything. My E. coli is not quite the same as your E. coli, in the same sense that my genome is not quite the same as your genome. And the degree to which our microbial compositions and their genomes differ is actually, as we'll see in the next few slides, much greater than the degree to which our genomes, or human genomes, differ. And one of the ways in which we can see this is looking at barcodes of individual microbial markers. What we're doing in this heat map is unrolling not the relative abundances of many different microbes in a selection of metagenomes, but the presence or absence of individual unique marker sequences within one microbe, using Prevotella copri as an example, in a series of metagenomes. So we've unrolled along the y-axis here the Prevotella copri genome. Each row corresponds to one unique marker sequence. This is a subset of a few dozen unique markers from a few hundred total in Prevotella copri that vary from individual to individual. And what you're seeing here is the gain and loss of genetic regions among different individuals' Prevotella copri strains. I mentioned at the beginning that our bugs have much more plastic genomes than we do. And there's a lot of gain and loss of particular loci. Here, what you can see is the persistence of strains with particular genetic configurations over time. The top bar here is colored by subject. from whom we have either 2 or 3 sample metagenome samples drawn over time. And these are stool metagenomes representing the gut, in contrast to the previous slide's skin metagenomes. And within, for example, the first individual on the left here, you can see a particular barcode of locus losses and gains in Prevotella copri that's persistent over time. And there is a small degree of change. You can see 1 or 2 loci that change. These samples are drawn about 6 months to a year apart. But to a very good approximation, everyone has a slightly different strain in this population of Prevotella copri, and that strain is largely stable within individuals over time. So you can zoom in and quantify not just the abundance of species within a metagenome, but unroll these marker barcodes to identify and track the abundance of particular strains in individuals. So this has both functional consequences, as we'll see in the second part of the talk, but also some interesting ethical implications, in as much as we can tell each other apart using, say, DNA forensics. Can we tell each other apart using microbial DNA forensics? If you look back over the very, very long history of about 10 years now of human microbiome work, almost every paper that you'll see has a plot that looks something like this. These are ordinations. They're similar to, for example, PCA plots, in which the overall similarity of 2 microbial communities is reflected by how close their points appear on the 2-dimensional projection. And the message that these kind of ordinations were used to convey for many years now is everybody is a little different. Every microbial community is a little different. It's very easy to tell samples drawn, for example, from one source, one individual, one location, one material type, one medium type, from another. And some of this is due to the fact that, to a good approximation, our human genomes, especially as far as coding nucleotides, are all mostly the same. And there's a very small fraction of, admittedly very important, diversity in the human genome. To a good approximation, our microbial communities are, again, mostly different. It's much, and these are real data from the Human Microbiome Project. It's not atypical for 2 individuals' gut microbial communities to share 10 to 20% of their organisms, let alone genetic diversity within those organisms. And that's in the absence of perturbations from disease, for example. So those tiny variants in our human DNA are enough to be able to tell individuals apart, for example, through DNA forensics. What about these huge variants in our microbial community DNA and composition? So this is the question that Eric Franzosa, a current postdoc in the lab, started with a year or 2 ago, to be able to determine whether these microbial differences are enough to uniquely and stably differentiate individuals over time, raising both some opportunities as far as microbial tracking and some questions that are some policy questions that are similar to those that have been looked at for the past several years in terms of human DNA. So the question is not new per se. There is some very early work. One paper I particularly like is the second one listed here, in which NASA realized that there were different microbes taken to space and back by different members of early space missions. But in the intervening 30 or 40 years, this has been refined, particularly in the past few years, by the concept of microbial source tracking, because microbial samples do tend to differ really dramatically in their composition from person to person or source to source. This initial paper with 3 individuals a few years ago now suggested that, hey, everyone's different. What can we do with that as a starting point? The Human Microbiome Project observed the microbial genetic variation that I showed a few slides ago, in as much as different individuals in a population will have microbes that gain and lose specific genetic loci with a great degree of individuality and plasticity. Again, even more so than the human genome. And then more recently, Jeff Gordon's lab did a great study of exactly how stable strains are over time. I showed some qualitative information on the stability of microbial strains for up to about a year. Jeff's lab went in and put some numbers on this and identified the decay rates or the modification rates, or essentially sweep rates, of different microbial groups in the gut in particular over time. So we have some evidence based on this work and our own data that bugs can persist at the strain level within individuals for at least several years. Again, raising this question of exactly how uniquely can we tell individuals apart using microbial composition or microbial DNA sequences, and for how long? Because our bugs can and do change over time, even given this relative stability, much more so than, for example, our own genome does. So in order to formalize this question, Eric put together the concept of combinations of sets of microbial features, or metaomic fingerprints, where a feature can be the presence or absence or abundance of a particular microbe, or of a particular microbial gene or microbial polymorphism. So for the next few slides, when I say features, a metaomic fingerprint can be composed of combinations of any of these three things. And you can think of a microbial taxon as a taxonomic profile, just like we saw for the last few slides, or equivalently, the presence or absence of a whole microbial genome. We can use individual marker genes, just like we did within Metaflan, to derive taxonomic profiles, and that's then the presence or absence or abundance of a specific gene within a genome. Or we can look at individual polymorphisms, and since we don't typically, again, have the data to resolve this within microbial populations, you'll see these captured not as individual loci, but as kilobase windows, where we aggregate the polymorphisms that we detect over a sliding window inside of the genome. Any of these can be combined using, you can imagine, a number of different algorithms in order to differentiate individuals and test how long a particular set of features is able to differentiate individuals over time. So this was very exciting from a computer science perspective, because it turns out this is one of those rare cases in which you can nicely formalize a biological problem as a very well-studied classical computational problem. And in this case, it's an instance of what's called the hitting set problem, or equivalently the vertex cover problem. And so to define this in terms of sets, if you think about what's necessary to differentiate my microbiome from everybody else's, if this box represents all of the features, again, be they bugs or microbial genes or microbial polymorphisms, all of the features that are present in my microbiome, where I have highlighted the subsets of features that are not present in somebody else's microbiome. A combination of features that describe me uniquely are those that are sufficient, the three red ones highlighted here, to include every set. Hence the hitting set problem. I need at least one feature within each of these other sets to define my code uniquely. And I won't talk about it today, but this is, of course, a fragile code. You can make codes that are more robust, but as a baseline, the hitting set, or again, equivalently the vertex cover problem, gives us a way of defining an initial unique code. So this is great in that it's a very well understood, closely studied computational problem. This is bad in that it's very well understood and closely studied because it's one of the original 20-odd NP hard problems. So we know that we're not, in the general case, going to be able to get an efficient solution to this, especially in something very high dimensional like lists of microbial polymorphisms. But we also know that there are good greedy approximation algorithms available for hitting set. So there's a known a greedy 2 approximation that means it's at worst twice as bad as the best solution. So that suggests that if we define a good greedy criterion, i.e. a way to prioritize features, bugs or genes or polymorphisms, then we can plug that into the existing greedy algorithm for solving hitting set and derive unique codes for each individual. So what Eric did next is try to define a good prioritization criterion, a way to greedily choose features, bugs or genes or polymorphisms. So the first criterion that he looked at was simply temporal stability. So looking over an increasingly long sampling interval between a few months up to about a year, how stable were individual features, either microbes as measured by amplicon sequencing, microbes as measured by metagenome sequencing, individual microbial marker genes from shotgun metagenome sequencing, or again, the kilobase windows aggregating polymorphisms like I mentioned earlier. When I show these data, these are all from the Human Microbiome Project. These are a set of samples drawn from a total of 18 different body sites, so we can assess code uniqueness and stability throughout the body, not just in the gut. These are a set of samples that were sequenced metagenomically on a smaller subset, both of individuals and of body sites. And I'm happy to take questions about the Human Microbiome Project currently offline later as well. So this is a particular application of the amplicon and shotgun data from the project. When
At the end, when we're talking about stability, these people, like, they're not taking antibiotics, their diet is not changing.
Oh, it's a great question. So the question was how stable was the environment of the individuals in the Human Microbiome Project? The short version there is that at recruitment, they were required to be very healthy, not on medications, not have taken antibiotics in the past, I think it was 9 months. But between time points, and these are assessing, again, up to 3 time points of about a year between individuals, between time points, they could take antibiotics. The restrictions were lifted. About 15 or 20 of them did. We don't, to give away the punchline there, we don't see a difference in stability between individuals who did and didn't, but it's very small numbers because it's 15 or 20 out of 100 to 300. Diet, we don't know. So as far as identifying microbial features that were good at stably differentiating individuals, just looking for microbes that are persistent over time is a good starting place. But they weren't particularly unique among individuals, and there was only a relatively weak subset of the total microbial community that was stable over time. So you can see here that the GI tract, the mucosal surfaces, are a little more protected. As we mentioned earlier in the Jeff Gordon paper, the gut community in particular tends to be stable for up to several years, but it's a subset of that community that's stable. It's not particularly unique per individual. And if you're a bug, once you are there, if you have persisted for a few weeks to a month, you're probably going to be a longer-term persister as well. So there's not a big difference over time of which subsets of the community are stable. If we instead use microbial or gene prevalence as a criterion for greedy prioritization, if you are a prevalent microbe, if you are present in a large fraction of the population, you're likely to be there again in the future. And so this is a nice ecological property. If you're a bug that's popular in a population, if one individual loses you, you will tend to come back again in that individual later on. Or if one individual has you, you will tend to be present there again later on. But if you stop and think about for a second about the problem of using feature prevalence as a criterion, it's great for stability. It's terrible for uniqueness, because if you are a prevalent bug or feature, then everyone's going to tend to have you. So what ended up working really well is kind of a combination of these things, is specifically differential feature abundance. So if we look at and prioritize features by the difference in microbial or gene abundance between an individual and the rest of the population, we see both a very strong tendency for differentially abundant organisms to be stable over time. We see this segregate with the degree of differential abundance, and we see some segregation again by body site as well. So we're picking up temporal stability, we're picking up differential ecology, and we're picking up uniqueness. And so using this criterion, oh no, we have our one loss to the transfer of the presentation. Pretend there's a formula in that blank spot right there. It's not quite showing code, Mike, but it's close. This is the actual instantiation of the greedy algorithm with what's missing right here is essentially differential abundance as our optimization criterion. Features that are inserted into a metaomic fingerprint for an individual are those that are highly differentially abundant between that individual and anyone else in the rest of the population. So to translate that, if you have a bug that is really abundant in you and nobody else, that will tend to be a good marker for you, and it will tend to be persistent over time. And we have a little bit of additional steps inserted into the baseline greedy algorithm to help increase the code robustness. One of these tends to select features that are orthogonal from each other, that are different from each other. If I have one marker from a bug, I don't need another marker from the same bug. And another one tends to inflate the size of the metaomic fingerprints a little more than they would need to be. And again, that helps to trade off sensitivity for specificity, or excuse me, specificity for sensitivity. It helps to increase our robustness. So in order to assess sensitivity and specificity and robustness of our ability to identify individuals using these codes, Eric put together a set of definitions for false positive and false negative detections using the repeated samples from the HMP population. So if we have a baseline temporal sample from a set of individuals in the population, in some cases, at a future sample, we will be able to uniquely and correctly identify an individual using their code as derived at time 0. So hopefully in a large fraction of the cases. In some fraction of cases, however, we'll identify a false negative at a subsequent time point in that we can no longer detect somebody because a component of their fingerprint is no longer present. A gene has been lost or a bug has been lost from the community, so the fingerprint no longer triggers for that individual. And in an even worse case, in some ways, we can get a false positive detection at future time points if a code changes in such a way as to now match the incorrect individual. So if we gain and lose a bug, for example, so that a combination matches the wrong person. So this is our definition of false positives and false negatives that we put together to assess our accuracy in the Human Microbiome Project population. We have some additional evaluations that we did in a second wave of HMP sequencing and in the independent MetaHit project that all generated about the same numbers. So using just the initial set of HMP data over up to 3 time points, we first assess the degree to which, for each feature type of interest, you can make unique codes at time 0 or not. And it turns out that using amplicon taxonomic profiling, or especially using shotgun taxonomic profiling, your alphabet size is often not big enough. You don't have enough diversity to even tell individuals apart at time 0. Too many people tend to carry the same small subset of bugs, for example, such that even though our overall communities differ among individuals, there aren't enough unique individual organisms to build a code. But that wasn't true when we increase our alphabet size by using individual marker genes within bugs rather than whole microbial genomes. And this tends to be for that reason that I showed earlier, that within each bug, there's a subset of loci that are highly variable between individuals. So we can take advantage of that here. Kilobase windows I don't show here because they behaved about the same as marker genes, but with more noise. So they're not especially interesting. If we break down now the cases in which we can make unique time 0 fingerprints into those in which we subsequently detected the right individual at future time points, or lost an individual, or picked up a false positive of some sort. You can see that even in the cases, especially for amplicon data here, where you could derive a unique code at time 0, they're easy to be lost at future time points. So there tends to be a little more noise in amplicon sequencing data in the first place. There's a PCR step here that's a little more noise prone than shotgun sequencing. And again, the alphabet is small, so your codes tend to be more fragile. In our marker gene sequencing, we get very few false positives, and to be fair, we have tuned the target code size to prefer false negatives to false positives. And if you look from microbial community to microbial community across body sites, the gut in particular, as we started out observing in the Jeff Gordon paper, tends to be fairly protected. Microbes are unique and persistent there, and we can get uniquely identifiable codes that apply for up to about a year in 75 to 80% of individuals. This goes down to maybe 50% of individuals in this particular population for other protected areas of the GI tract. And then for the least protected areas, such as skin, where even at the anterior nares, which is the skin at the front of the nostrils, we see similar behavior for the forearm skin, which was also sampled in the project. You're down to about 25 to a third of individuals for which you can build and retain unique codes because presumably the community is more disrupted over time. We don't actually, as I mentioned earlier, we don't actually see a huge effect of time interval. It's slightly statistically significant in a few cases, but not very much so. And again, this is presumably because of what I mentioned earlier. If you are a bug that persists for a few weeks, you are probably going to persist for a few months or a year as well. And this was very concordant with what was observed in the gut earlier on. I think for the interest of time, I'll skip over the biological explanations of some of these codes. It boils down to different microbial ecologies in different body sites explain our ability to build codes. Sometimes it's due to strain level variants, such as in the vaginal microbiome. Sometimes it's due to species level variants in the gut community. And other times it's due to overall microbial diversity. The oral cavity, for example, just plain has a bunch of different bugs. So we can really pick out different bugs rather than different species, rather than different strains. So the upshot here is that individual identifiability using microbial composition, or more specifically microbial gene composition, is possible, typically from shotgun sequencing data. It's very challenging from amplicon sequencing for a couple of different reasons. It's accurate up to about 80% of the time in the gut, which is the most protected, most stable community that we and others have observed. And this is true in our population of 300 individuals. Like I said, we've validated this in external populations. The numbers are about the same. But more often, we get 50% to maybe 25% of individuals being identifiable over months to years because of the higher turnover and more change that's seen in other communities. It's minimally time sensitive. If you're there for a few weeks or a few months as a bug, you're probably going to be there for a year. And in this case, we built our fingerprints, our codes, using a greedy algorithm that was an approximation of the hitting set problem by prioritizing differential abundance. So this really gives a baseline. There's nothing saying that this is the best algorithm for identifying individuals using microbial community features. But this is a baseline, a starting point, from which you could build more accurate or more stable algorithms as well. So I'll actually stop and ask whether there's any questions here, because I'll talk about functional rather than taxonomic profiling, which is what we've been looking at for the last few minutes of the talk.
So what's the size population of
That's one of my supplemental slides. If you reviewed the paper, one of the reviewers asked that too. It's scale, so human DNA identification, the earliest technologies scaled to about tens of thousands of individuals. Current technologies scale to billions. It's very easy to get very precise human DNA measurements. When we evaluated this, we get hundreds to thousands of individuals. So the current coding strategy will apply up to 500 to 1,500 individuals. So it is not unique out to, say, a billion individuals as per very precise RIFLET measurements, for example, for human DNA forensics.
Still won't replace DNA sequencing.
It's not nearly as accurate either. And I could say this at the end instead, but to insert it here, honestly, the practicality of this for individual identification of individuals, I think, is not super exciting. But there are entities such as locations that have microbial DNA profiles that do not have their own intrinsic DNA profiles, such as individuals. So I'm very interested to see whether this scales to locations or to determining whether individuals have been in a location, since that seems like a more practical application than just saying, hey, you can tell people apart. Where I think this has more direct implications is how we handle and distribute and treat microbial community data. Because human DNA data is very tightly controlled by NHGRI, by dbGaP. There are zero restrictions on microbial sequence data sharing right now. And I'm not sure I'm entirely comfortable with that. So hopefully this will at least be a reason to think about that kind of data privacy a little bit more.
I think you answered my question. Like, usually when you want to identify people, those people are trying to put down genes. You're not worried about that. You're not worried about that application. You're not trying to identify criminals. You might be trying to spoof.
We have not thought about microbial community spoofing yet, no. Although it's a really interesting idea. There is the question of if you shake somebody's hand, can you tell whose it was and for what period of time? And again, we have no data on that, but it's a neat follow-up question. Yep.
this to the microphone? Yeah.
So I guess the Human Microbiome Project was done with these sort of unusually healthy people. And I guess, a healthy gut is usually, or any surface is sort of generalized by being very diverse. So I'm wondering whether your sort of analysis would break down when you think about, all the unhealthy people who have a less diverse microbiome, if you will.
That's an interesting question. It's rare to see a really dramatic decrease in diversity. In the second part of the talk, I'll talk a little bit about inflammatory bowel disease, which one of the earliest observations is that it's characterized by a decrease in diversity. But that can mean a decrease from, say, low hundreds of species per individual to 1 to 200 species per individual. So the decrease in diversity, unless you're talking about something that's an acute infection, so like Clostridium difficile infection, the decrease in diversity is modest rather than dramatic. Even in those cases, I'll actually zoom back to here, the vaginal community is interesting in that it is, in health, very low diversity. It's typical to have one or, in many individuals, to have one or a few lactobacillus strains per individual. And so what the algorithm ends up doing is actually detecting that and using more markers to tell apart individual strains rather than using fewer markers but picking out multiple different species. So just a lower level of ecological diversity doesn't necessarily hurt you in as much as you can still zoom into the strain level to tell things apart. So in the last few minutes, that actually gives a good segue to discussing functional profiling and a quick application in some work that we're doing on inflammatory bowel disease by focusing not on microbial composition, but on microbial metabolic or functional composition. So rather than who's there, what are they doing? And one of the reasons that we really started looking closely at this a few years ago is that in one view of this diversity that you can see here along the top, this is a subset of our individuals from the Human Microbiome Project again, with the composition in each column highlighted at the whole phylum level by color. So if we focus just on the gut, for example, these are 100 or so individuals, some of whom have guts composed almost entirely of blue, which is Bacteroidetes in this case, some of which have guts composed almost entirely of purple, which is Firmicutes. And these are the healthy folks, again, those unusually healthy individuals. And so this is why I highlight how much diversity, how many differences there really tend to be in microbial composition between individuals. It's true not just in the gut, but in pretty much any human-associated microbial environment that we or others have surveyed. But you can contrast this to the relative stability of the distribution of microbial functions within communities. So what you're looking at here on the bottom is the same samples, the same microbial communities, the same data from shotgun metagenomes, classified not into which microbe each sequence came from, but into which broad pathway it came from. And so, of course, some of the most common or most abundant pathways are housekeeping, are the ribosome, or the cell cycle, or central metabolism. But there's a core of less housekeeping, less basic pathways that are unique to each microbial habitat and are stable in their relative abundance between individuals, at least in proportion to the instability or the differences in which microbes are carrying them. So a way in which I like to think of this is about the jobs within a city. You have different people with different names living in different cities, but each city has some distribution of economic functions or transport functions or banking functions or academic functions that are taken by the roles that are taken by different individuals in different cities. So here we have a distribution of roles that need to be filled in a particular environment that's taken on by different microbes in different ecologies. So this has been observed not just in the Human Microbiome Project, but in several other cohorts as well. Pretty much any microbial environment where you see taxonomic diversity, if there is functional consistency in the ecology, you can see this kind of stability. And it's nicely explained. I like this paper because it makes the same observation not about the human microbiome, but about algae-associated microbial communities. And this is nicely explained by some of that genome plasticity that I mentioned earlier. Microbes have access to mechanisms such as lateral transfer that allow them to balance their functional repertoires on a much more rapid timescale than, for example, human genetics could account for. So this is a great representation of a core of the human microbiome, or properties of the microbiome that we all share. And it's really interesting, again, computationally, because there are not assigned functional roles for a very large fraction, depending on how you count, about 50 to 75% of the microbial genes that we observe in the human microbiome are not functionally annotated. So there will undoubtedly be more functional diversity than we can currently see among these unannotated genes. But it will probably also speak to some of the really interesting signaling and ecological functions in addition to sort of basic metabolism in these communities. So I'll skip quickly through some of the methods that we've derived for functional profiling in metagenomes and metatranscriptomes. One very targeted method is called shortbread. This is a functional profiling tool that uses the same intuition as metaflan. By finding short peptide sequences that are unique markers for functional gene families, shortbread can rapidly profile very targeted gene sets in the same way that metaflan can very rapidly profile very targeted taxonomic clades. So that's the one slide overview of the algorithm. More importantly, to give an example of the information that shortbread can derive from a collection, again, of metagenomes, of shotgun metagenomes in this case. What you're looking at is a set of markers, unique peptide markers for antibiotic resistance gene families, derived from the Antibiotic Resistance Database, or ARDB, by a master's student in the lab, Jim Kaminsky, and then classified into the broad antibiotic resistance families that ARDB defines, such that for a collection of metagenomes surveying both the Human Microbiome Project, a set of Chinese gut metagenomes published as part of a type 2 diabetes project, and a series of global metagenomes from the United States, Venezuela, and Malawi that were sequenced to a lower depth, if you're wondering why this section is a little less dense than the others. We can derive very specific markers for individual antibiotic resistance gene families and profile out not which bugs are there, but which gene families are there in what relative abundances. And this was work with Gautam Dantas and his student Molly Gibson at Washington University. More generally, for larger scale or broader, less targeted functional profiling, we've recently completed work on the second version of HUMON, which parallels metaflan as a functional profiling rather than taxonomic profiling too. And the very quick overview of how HUMON-2 works is to begin with the same set of genomes that Nicola derived earlier from the NCBI for use in metaflan and our other gene cataloging efforts. So this is where we get our 17,000 or so total bacterial and other genomes. To give a few sources some bad press here, we used to use IMG instead until they became no longer publicly available. So that was a problem for us and many other people. From our genome set that's now derived from the NCBI, Nicola creates the Chocoflan catalog that I mentioned earlier that organizes annotated genes into gene families. These are exactly what we use to derive unique taxonomic markers from metaflan, and we organize the annotations, the functional information and pathway annotations in these genes for use in functional profiling in HUMON-2, where the very quick overview of its hierarchical process is to take a metagenome or metatranscriptome and identify which organisms are most abundant using a metaflan2 prescreen. So very quickly say which bugs are there. Take just their pangenomes annotated within these gene families, make that a targeted, very specific gene database, and rapidly map reads to it at the nucleotide level using Bowtie2. For reads that do not hit this targeted gene database, then perform a more expensive translated search of a larger gene catalog using Diamond, which is a recent accelerated translated BLAST alternative. And finally, counting hits to individual gene families, these are reassembled into pathways using either Metapsych or Unipathways pathway definitions. And so both of these are catalogs that associate genes with pathways with their potential metabolic products or reactants. This is another case in which we used to use KEGG information. Since we started working with Human, KEGG has also become no longer publicly available. So both of these were cases in which we had to make fairly major shifts in our data sources because a resource suddenly disappeared. All of this, both the software and the data that we use for Human 2 now are publicly available. And I should have said this earlier, anywhere you see one of these URLs, there's some combination of software, documentation, tutorials, downloads, and for the tools that are already published, the paper associated with them. So this is a quick overview of how Human 2 works. What it provides for you at the end is a profile of which genes and pathways are abundant in a sample. And to wrap up the talk, I'll give a quick example of how we've been using that to get a better understanding of functional dysbioses in inflammatory bowel disease. So as a very brief crash course in the microbiome in inflammatory bowel disease, or IBD, IBD has become one of the poster children for complex polymicrobial microbiome-linked disease, in as much as starting about 5 or 10 years ago now, some of the earliest observations of ecological disturbances in the gut during IBD were made. So there was a reduction in diversity, fewer bugs present in the gut of individuals with Crohn's disease or ulcerative colitis, the two major subsets of IBD. And then over the subsequent few years, this was refined into observations of specific clades that were more or less abundant in the guts of IBD patients. And then finally refined into some heterogeneity within disease subsets. So there are certain intestinal localizations of Crohn's disease, for example, that tend to be etiologically more severe, that have distinct microbial dysbioses as well. And we've gone on to describe some of the microbial metabolic functions that correspond with these overall ecological disturbances. And I'll describe this in a few slides. Starting about a year or two ago now, we wanted to organize a more detailed functional description of what types of actionable microbial metabolic processes are disturbed during IBD. And some of the ways that we wanted to target this in a way that would be clinically useful was to better understand patient heterogeneity within these diseases. So both within Crohn's and within ulcerative colitis, some individuals respond well to first-line treatments and some do not. Some individuals have relatively quiescent disease over time and some do not. There's a lot of patient heterogeneity. And so the microbiome provides potentially a new high-dimensional readout that could be used to understand population stratification within IBD. Similarly, if we look at longitudinal disease activity over time, both Crohn's and colitis tend to operate in flares, such that there will be periods of, within each individual, periods of relative quiescence, followed by periods of disease activity, which then resolve into quiescence again. So if we could either predict the onset or resolution of flares using a high-dimensional microbial readout, that would allow us again to better target treatment of those individuals' disease activities. And finally, both on the microbial and the host side, there's the opportunity to identify new bioactive small molecules or targetable pathways for improved treatments in IBD. As there is, beyond first-line treatments, there's a relatively small number of treatment options available. So we're about halfway through a project now that's part of the Human Microbiome Project 2, or HMP2 umbrella, which specifically has the mandate to build a better multi-omic functional understanding of disease overall. There are two other projects on maternal child health and type 2 diabetes, and then ours, of course, looking at Crohn's and colitis. So I won't go into the details of our sample collection and data generation process in the interest of time. As the slide probably conveys, we're really hitting a collection of samples with the kitchen sink as far as data generation. This was part of the NIH mandate. For about 2,000 of our primary samples, which are stool samples collected every other week from our longitudinally tracked patient population, we had a whole set of mostly sequencing-based microbial assays, ranging from amplicon and metagenome sequencing through metatranscriptomes to metabolomics and proteomics. And from a series of primarily host samples, biopsies and blood draws, we get genetic information in addition to some host gene expression and epigenetics as well. We, of course, can't get the biopsies quite as longitudinally over time. We get those at recruitment and at the 1-year patient exit, and we get quarterly blood draws. So the upshot is that this project is deriving a wide range of high-throughput molecular information, profiling bugs and the host longitudinally in just under 100 individuals in Crohn's, colitis, and controls. So we've done a validation study to build a much smaller and more convenient fixative-based kit for mailing in stool samples that's compatible with these multiple different types of data generation. If you're interested in this, ask me about it offline after the talk. The entire protocol is available online if you want kit numbers, assembly instructions, things like that. And we have a series of web portals that we've been developing both for rapid data release for the HMP2 project. So this includes a series of automated workflows that quality control and process and post to the web newly generated data. These include some pilot data that we have now. Again, we're about halfway through the project, so of course the entire data set isn't available. But we have overview visualizations and quality control information, processed data products, and raw data files that are automatically detected upon generation, processed using a reproducible workflow, and posted through an HTML template to the web. And so there is some pilot data available through the project already as well, some of which is being returned to subjects. So we have IRB permission to return aggregate data to participants in the project, which really helps in keeping them enrolled over a year's worth of biweekly stool sampling. So this allows them to see what their microbial configurations, what their most recent microbial time courses, and their average microbial and dietary profiles, which is some of the metadata that we have available for the project, look like in comparison to the broader population. So finally, to wrap things up, we know from earlier cross-sectional studies that there are consistent microbial and microbial functional differences between Crohn's and colitis patients cross-sectionally, in terms of a broader population, and healthy controls. And so to start using some of our pilot data from the HMP2 to zoom in on the potential metabolic mediators of these differences, what we've looked at is a series of pilot samples from a cross-sectional subset of the total target population, a very small longitudinal subset of the population that we had available as of a few months ago, and an independent validation cohort from the collaborator in the Netherlands, Rins Wiersma, from which we had very similar data from a small number of individuals to use as validation. And these data include shotgun metagenomic sequencing for a total of about 250 samples, some cross-sectional and some longitudinal, and metabolomic profiling. And again, to keep things short in the interest of time, our metabolomic profiling is untargeted. It includes about 400 known metabolites and initially about 70,000 unlabeled metabolite peaks. If we throw a linear model at this to test for differential abundance in Crohn's or colitis patients, we're able to detect just over 1,300 differentially abundant metabolites, about 60 of which are labeled, which we can then associate with our shotgun metagenomes and our microbial composition and function. These include several known bile acids in particular that are known to be disrupted in inflammatory bowel disease. Bile acids are microbially processed gut metabolites, some of which are either enriched or depleted during Crohn's disease and ulcerative colitis. And we'll use that as our last example in just a few slides. And we can look at this information temporally as well, and I'll actually skip this in the interest of time to return to our cross-sectional analysis. So one of the things that I'm most interested in this particular cohort is identifying small molecules that are likely to be either causally or consequentially involved in Crohn's and colitis that are microbially processed either as metabolic products or reactants by specific organisms. And we can start to identify candidates for these criteria now by looking at longitudinal information to help us get an idea of causality, and microbial correlates and genome gazing to get an idea of potential metabolic links between small molecules and bugs. So what you're looking at here is correlations between microbes that are differentially abundant from our taxonomic profiling in Crohn's and colitis, and metabolites that are differentially abundant in Crohn's or colitis. And within each of the overall block structures, here we have bugs and metabolites that are up together. Here we have bugs and metabolites that are down together. Within each of these large blocks, you get emerging groups of related metabolites and bugs that we can then look into the microbial genomes to say, hey, are these metabolites things that those bugs might be consuming based on their annotated pathways, or things that they might be producing? And to wrap up with the case of bile acids in particular, if we pull out just a single bile acid hydrolase gene family, so this is one of the gene families that HUMON2 knows about that we can place into the bile acid processing pathway, HUMON2 provides organism-specific detections. So each row is one bile acid hydrolase gene family in one organism, which we can then test across our samples to detect those that are differentially abundant, either enriched or depleted in inflammatory bowel disease. And what we see here in this initial data are a trade-off between some of the bugs that are known to be protective or depleted in Crohn's and colitis, and some of the bugs that are known to be enriched. And this starts to provide a hint as to potentially why, that the differentially abundant bile acids may be a mediating molecule such that we're trading off protective organisms that generate one subset of bile acids using a particular instance of the bile acid hydrolase family for risk-inducing or less, more dangerous or less friendly bugs that generate a different family of bile salts using a slightly different bile acid hydrolase. So to wrap things up, the tools that I mentioned today are all available on the lab website, again, along with documentation and hopefully tutorials and comprehensible example data when possible under the URL here. The BioBakery also includes a virtual image of pre-installed tools so that you can download them and get things up and running with minimal fuss. And there are a lot of great areas in which there's still ongoing work in the microbiome, ranging from very targeted microbiology or immunological mechanisms, all the way up through microbial ecology or human ecology in the sense of epidemiology. And I won't go into the details now since I'm horribly over time, but if you're interested in working on the microbiome or already working on the microbiome, we've only been doing this for about 5 or 10 years now, and there are a lot of great open questions in pretty much any area of biology, ranging from very detailed molecular mechanisms up to policy questions, for example, that might be derived from areas such as microbial identifiability that we've talked about today. So I want to thank the folks in my lab that worked on several of these projects, and especially our collaborators, Gautam, who we worked with on shortbread to identify antibiotic resistance gene families, Catherine Lemon and Brendan Bohannon, who were involved in the identifiability project, and Rob Knight, Romnick Xavier, and Dirk Evers, who've contributed to just about everything that I've talked about today. And thanks again to everyone here today.